So next part is internal structure of anther. Internal structure of anther. So anther contains mainly two parts here. One is called as anther wall and other is called as a sporogenous tissue. Two parts are there. One is anther wall and second one is a sporogenous tissue. Okay, before going to this internal structure or the parts that we are going to discuss here, so one thing we have to know that in the dithecous and monothecous, so if you take the dithecous condition, as you said that two anther lobes are present. Each anther lobe contains two pollen sacs, or pollen sac is otherwise called as microsporangium. So that total 4 microsporangia are present, or 4 pollen sacs or pollen chambers are present. As 4 microsporangia are present, we call it as a tetrasporangiate condition. Tetrasporangiate condition. Whereas if you come to the monothecous anthers, so as I said that monothecous contains only one anther lobe and each anther lobe contains 2 microsporangia, so that total number will be here only 2. So we consider this as a bisporangiate condition. So this is a variation that we can see based on the number of anther lobes. So dithecous is tetrasporangiate, whereas a monothecous is bisporangiate in condition. Okay, as we are discussing in the internal structure of anther, two parts are there. One is anther wall and other is a sporogenous tissue. So before that, we should know something, that anther wall is a protective structure. Mainly it is concerned with protection of the inner tissue, and some layers of the anther wall are also helping in the dehiscence of anther. So when anther is breaking, the pollen grains are released, which are transferred to the other flowers or the stigma of the other flowers or the same flower, that is helping in the pollination process. So here let's come to the anther wall. So here anther contains mainly 4 layers. Anther wall contains mainly 4 layers. So this is the outer view of the anther in the transverse section, where we can see 4 lobed structure. If you take the transverse section of the anther, we can see this type of outer view in which the anther wall is present. This half part is called as one anther lobe and this second part is called as a second anther lobe. So this is a transverse section of the dithecous anther. In this one, the middle part is there, which is considered to be the connective like structure. Okay, and each anther lobe contains 2 pollen sacs or microsporangia like this. So that in total 4 microsporangia are present. And let's come to the first anther wall. Each anther wall is differentiated. Actually, anther wall is of 3 to 5 layered. So which is differentiated into different parts. The outermost one is called as a epidermis like this. So which is considered to be the outermost layer and the main protective structure. The main protective structure of the anther wall is the epidermis. So which is outermost layer. So the same type of organization can be seen on other side also. So this is called as epidermis. And this epidermis is mainly made up of parenchymatous tissue and is concerned with protection. Below this, there is one more layer is present, that is called as endothecium. This layer is called as endothecium, which is considered to be the hypodermal. So why we are considered to be the hypodermal layer means any layer which is present just below the epidermis, we consider it as a hypodermis or hypodermal layer. As this endothelium is present just inner side to epidermis, we consider it as a hypodermal layer. It is also made up of parenchyma cells, but a little deviation is there between the endothelium cells and the epidermis cells. That is, endothelium cells contains the fibrous thickenings on their walls. Fibrous thickenings, elongated fibrous thickenings are there, which are made up of alpha cellulose. And because of these fibrous thickenings, they are acting as hygroscopic, or they exhibit hygroscopic nature. So what do you mean by this hygroscopic nature? Means depending upon the moisture that is present in the environment, so the cells may contract and also expand. While they are contracting and expanding based on the availability of moisture, they are helping indirectly in the breaking of the anther wall. So that endothelium is helping in dehiscence of anther and releasing of pollen grains. Okay. So this is one part which is helping the dehiscence. And in epidermis also there are some specialized thin walled large size cells are present. Those are called as stomium cells. So this is the epidermis as we have said here. So let's come this side so it will be very much clear. So in the epidermis part, between the two pollen sacs, so some of the epidermal cells become larger and thin walled. So these larger and thin walled cells are called as stomium cells. These are the part of epidermis which are present between the two pollen sacs or between the microsporangium of one anther lobe. So the same can be seen on this side and also on this side between the two pollen sacs. So these are also helping in the dehiscence of anther wall and releasing of pollen grains. So here endothelium and also the stomium of epidermis, both of them are helping in the dehiscence of anther wall and releasing of pollen grains. So next layers are called as a middle layers which are present inner side to the endothelium. So these are the middle layers. Just below the endothelium layers, we can see one more layer or the third layer of cells in the anther wall. Those are called as middle layers. Middle layers are also thin walled cells as like the stomium cells of epidermis, and these are generally 1, 2, 3 layered. And actually some scientists they consider that there is no specific function related to the middle layers, but they are also considered to be the nourishment tissue. Nourishment means they provide food materials to the microspore mother cells or the microspores which are present inside the pollen sacs. So they are considered to be the nourishing tissue or the food material providing tissue for the microspore mother cells or the microspores. And the innermost layer is called as tapetum, which will encircle the sporogenous tissue which is present in the pollen sac. So this layer is called as a tapetum tissue. So here tapetum tissue have many special features. So this is single layered, and as like other layers it is also made up of parenchyma tissue, and these cells are also generally thin walled. And tapetum cells are containing cytoplasm which is dense cytoplasm. We can say the thick cytoplasmic contents are present. Along with that one, sometimes they may exhibit multinucleated condition, or sometimes what happens, instead of this multinucleated condition, so they may also show polyploidy. Generally, when I am saying about multinuclear condition, the number of nuclei may be ranging from 2 to many, or binucleated to multinucleated condition, but in most of the cases we say it as multinucleated only, that is cenocytic condition is present. and sometimes they may also exhibit the polyploidy condition. And apart from this one, the main functions related to tapetum are they are acting as a nutritive tissue to the developing microspore mother cells or the pollen grains which are present inside the pollen sacs. And apart from this nutritive tissue, or we can say that by the time the microspore mother cells are completely or converted into pollen grains, so the entire tapetum layer will be exhausted. Exhausted in sense it have been completely consumed by the developing pollen grains. And apart from this tapetum, as I said, that middle layers are also acting as a nutritive tissue. So that middle layers are also being completely exhausted, completely utilized by the developing pollen grains. So when the anther become matured or when it is ready to dehisce, at that time we can see the presence of only the epidermis. Single layer of epidermis can be clearly seen in the matured anther where it is ready to dehisce and release the pollen grains. And other functions related to the tapetum are, it is also helping in the formation of pollen kit, which is made up of proteins and carotenoids, which contains the proteins and carotenoids, and the pollen kit is the one which is also concerned with the formation of the sporopollenin. So this sporopollenin is a complex organic material which is the part which is a chemical present in the exine of the pollen grains. So that is also been formed by the tapetum cells, and it also helps in the formation of callase enzyme. So what is the function of this callase enzyme is, during the microsporogenesis process where microspore mother cell is converted into 4 microspores. Between that 4 microspores there is an callose wall formation is present. That is the enzyme will help in the breaking of that callose wall such that the 4 microspores are separated from each other. So these are the functions related to the tapetum layer. So the most significant one in this one is considered to be the tapetum layer. So next one is sporogenous tissue. So sporogenous tissue is a fertile tissue which is present inside the microsporangium or the pollen sac. So this sporogenous tissue is formed from the hypodermal tissue of the anther, that is the archisporium. That's why the development of microsporangium is called as eusporangiate type, and this sporogenous tissue is formed from the inner layers of the sporogenous tissue. So this sporogenous tissue is generally diploid in condition. And this sporogenous tissue, first initially it will undergo mitosis to form microspore mother cell. Or let's take each and every sporogenous cell, and that sporogenous cell will undergo mitosis to form pollen mother cells, or otherwise we can call them as a microspore mother cells. So these microspore mother cells or pollen mother cells, these are also diploid in condition because these are formed as a result of mitosis from the sporogenous cells. And this microspore mother cell, this is considered to be the precursor for the microspores, and it is a mother, in the name itself it is very much clear that it is a mother cell for the microspores. So that means the microspore mother cells will be converted into microspores in the process of the maturation of the anthers.